Welcome to day 2 of India Alliance's annual Fellows meeting. First off, we have a talk by, we start off the day with a talk by Professor Wendy Bickmore. Professor Bickmore is the director of MRC Human Genetics Unit at University of Edinburgh, and she's been the president of genetics of the Genetics Society since 2015. And most fortunately for all of us at India Alliance, she's a member of our Early Career Fellowship Selection Committee and the deputy chair of the committee. Her research involves multidisciplinary approaches using cytological, genetic, genomic, and biochemical methods, as well as animal models to understand genome spatial organization and how it contributes to gene regulation. Professor Bickmore will talk to us today about the remote control for gene expression. Professor Wendy Bickmore.
Thanks very much. It's wonderful to be here and fantastic being associated with the India Alliance. As you will see in my talk today, I've greatly benefited from the quality of PhD students who've been trained here in India. And in turn, I hope I will be able to return some postdoctoral scientists to India for fellowships, perhaps from India Alliance in the future. So I'm going to try and give you 3 short stories today from work in my laboratory that addresses one of the most interesting questions, I think, in mammalian genome regulation. How are our genes regulated in time and space, in development, in response to physiological stimuli, and how are they misregulated in disease? So the first story is about how we assign pathogenicity. So how do we determine whether a mutation is disease-causing when it doesn't lie in the coding region of a gene? And for those of you who are not familiar with the human genome, you should realize that most of our genome does not code for protein-coding genes. Most of it is non-coding, 98% of it. Whereas we have the same number of genes, roughly as a fruit fly, we have orders of magnitude more regulatory elements that control the expression of those genes, and we call these elements enhancers. We don't know how many enhancers there are in the human genome. We know how many genes there are. We don't know how many enhancers there are. Best guesstimates at the moment are around about 1 million or so. So that gives you something, tells you something about the complexity of gene expression. And here's 2 examples of some of my favorite loci in the human genome. The PAX6 and the sonic hedgehog locus. They're both genes that are very important in development. And so their expression in time and space during development is exquisitely controlled by sets of these non-coding enhancers. So this one here, PAX6, this is a gene that encodes a master transcription factor important for eye development. And we know so far of 35 different enhancers, these colored lollipops, that regulate the expression of this gene during embryonic development. These enhancers are spread over 500,000 base pairs of the human genome. And some of these elements are even embedded inside the introns of other genes that got nothing to do with the PAX6 gene. Here's the second locus here, sonic hedgehog. Its regulatory elements that we know about are spread over 1 million base pairs of the human genome, and again, often embedded within neighboring genes. So incredibly complex. So of course we know that mutations within protein-coding genes can cause Mendelian disorders in humans. And here's an example for the PAX6 gene. PAX6 gene is very important for eye development, and a haploinsufficiency of PAX6, so loss of the ability to make protein from one copy of this gene, produces a developmental disorder called aniridia, which is the absence of proper eye development, leading eventually to blindness. So of course, when we see a new patient with aniridia and we find a mutation within the coding region of this gene, it's fairly easy to decide pathogenicity because we know the genetic code. We know if a DNA sequence change changes the amino acid coding potential of the gene. But if it lies in a non-coding space, we don't know that. But we do know that mutations in enhancers and non-coding DNA can cause disease. So here's some examples here. Where we know from human genetics that mutations within enhancers cause severe Mendelian disease, just as severe as if there is a gene mutation itself. So So for example, of course, enhancers don't work if they're not there in the genome, so if they're deleted. So here's a few examples here. My favorite one here belongs to the SOX9 gene here. Small deletions, 1 million base pairs away from the SOX9 gene, produce a human syndrome called Pierre-Robin sequence. And this is because this mutation, 1 million base pairs away from SOX9, removes an enhancer which is required to activate SOX9 expression to drive chondrogenesis during development of the skeleton, particularly the long bones and also the face. So you end up with the cleft lip palate and bowing of the long bone syndrome. These elements are exquisitely dosage sensitive, so duplications of these elements also produce disease. Here's an example from the sonic hedgehog gene, the second one down here. The limb enhancer for sonic hedgehog. Again, 1 million base pairs away from the gene it's regulated. A duplication of that element causes a severe limb phenotype in humans. So these elements can work at great distance, but they are distance limited. So a chromosomal inversion that takes the enhancer and sends it further away down the chromosome also breaks the communication between a gene and its enhancer. And these enhancers work at great genomic distance, but only in cis, not in trans, so that a translocation that moves the enhancer onto a different chromosome from the gene it's supposed to control also disrupts function. So here's another example from Pierre-Robin sequence where the enhancer has not been deleted, it's been translocated onto a different chromosome. But also, we know that point mutations in these enhancers can produce disease, because enhancers are the place where the tissue-specific transcription factors bind. That's where the specificity is for gene regulation. So point mutations that disrupt transcription factor binding sites can prevent the target gene being regulated, but you can also have mutations that create new binding sites for transcription factors to give you improper gene regulation, so gain-of-function mutations. And even here an interesting one where we have an autoregulatory mutation, where the mutation is in enhancer for PAX6, the gene I just told you about, and it disrupts the binding site for PAX6, the product of the gene it's regulating. So this enhancer is required to sustain PAX6 expression over development. So that's all fine. So I'm going to show you an example of that. So this is this autoregulatory mutation here, in one of these blue lollipops here. And you can see here, so this is the phenotype of loss of function of the PAX6 coding region. So this is aniridia, the failure to develop an iris. And here's the phenotype when you have absolutely no mutation whatsoever in the PAX6 coding region, but a single nucleotide change with this enhancer over here, which stops PAX6 being expressed properly during eye development. So whereas we have enough information about protein coding genes, and the genetic code to be able to tell parents something about the cause of disease in their child, we don't have enough information about enhancers to be reliably communicate any useful information back to families who are affected by inherited disease where the mutation is enhancers. So we want to be able to fix that. We want to learn enough about how enhancers work to be able to incorporate mutations in this part of the genome into clinical genetic practice. And the person who's been leading this work in my lab is Shipra Bhatia, originally from India, and she's an early career fellow who I host in my lab. So what Shipra did was she went to our cohort of patients who have aniridia, and we excluded all those who have a mutation in the protein coding region, PAX6. We're not interested in those. We know why that causes disease. And we took the rest, the so-called exome-negative cohort of patients who have aniridia, and resequenced the enhancers of the PAX6 gene here. And already, this is still ongoing, already Shipra has identified 4 potential pathogenic mutations, de novo mutations, so they weren't present in the parents of the children. So there's good a priori evidence that these might be the pathogenic mutations and cause of the disease, but that's not good enough evidence in a clinical setting. So we need a bit more evidence before we do that. So as I told you, so we need some kind of experimental assay to say whether these enhancers are disrupted or not by the mutation. But I've already told you these enhancers only work in the context of development, so you can't do this in cell lines. You've got to do it in vivo. So that's the challenge Shipra set herself out to do. So what she's done is she's set up a relatively rapid in vivo functional assay for human regulatory variants using zebrafish, a really excellent model vertebrate. What she's done is engineered into the zebrafish genome a safe landing site where we know there's no position effects when we insert a particular cassette. And the cassette that Shipra has chosen to insert contains 2 reporters, one driving GFP, a promoter driving GFP, and a promoter driving cherry. In between them is a strong insulator, so there's no crosstalk between these 2 reporter cassettes. And then you can drop in to these 2 cassettes your wild-type version of the enhancer, and that might, for example, drive GFP. And on the other side of the cassette, you can drop in your suspected mutant enhancer, and that will be driving cherry. And you can introduce this into the zebrafish genome and then ask what's the regulatory potential of the wild-type enhancer versus the putative mutant enhancer. So here's an example of how well this works using that PAX6 aniridium mutation that disrupts the binding site for the PAX6 gene itself. So this is the expression pattern that the wild-type enhancer drives. So it comes on about 48 hours post-fertilization in zebrafish. It comes on in the lens and parts of the developing brain, the diencephalon and the hindbrain, indicated by D and HB there. And then by the next day, by 72 hours, that expression pattern has been refined to the lens only. So how does our suspect mutant enhancer behave in this assay? So this is now the assay using this dual color reporter. So we're reading out GFP and cherry expression in exactly the same cells of exactly the same developing zebrafish. So we get a really quantitative assay of enhancer activity. So on this side, on the left-hand side here, we put the wild type in red and the mutant in green. And on the right-hand side here, we've swapped the colors and we've let the wild type enhancer drive the GFP. And I hope you can see that in both cases, the lens of the developing fish is only being activated by the wild-type enhancer. There's no activity coming out of the mutant enhancer. So we think that gives us good enough information to be able to say, yes, this is a pathogenic mutation that is responsible for this disease. So we have now, I think, a functional assay where we can start for some developmental enhancers to provide useful clinical information back to the clinical genetics service. So that's all very well for Mendelian disease because, of course, we have very strong phenotypes there. Would this also be useful in other settings? So as probably many of you know, most of the genetic variation that's associated with common disease, so common variants in the population, that might link to diseases such as cardiovascular disease, diabetes, neurodegeneration, cancer. These GWAS hits don't lie in genes, they lie outside of genes, and most of them lie in enhancers, and they can lie in enhancers that are hundreds of thousands of kilobases away from the genes they control. So very likely these common variants subtly perturb the function of enhancers. They're unlikely to completely ablate enhancer function because that would produce a Mendelian disease, but they probably subtly affect their function. And here's an example why we think that actually Mendelian disease can be really useful for understanding these genome-wide association hits here. So this is not our work, this is something we found in the literature. Someone did a GWAS for eye phenotypes that are related to risk of glaucoma. And here's a particular trait they measure, which is the size of the optic cup. Here's your typical Manhattan plots that you're probably all familiar with from genome-wide association studies. And here there's a very strong peak here that in this paper they've associated with this gene called ELP4. Now ELP4 is part of, is involved in protein translation. It's a housekeeping gene. It seems unlikely that it's got much to do with the area of the optic cup development. But when we actually look at the lead SNPs for this study, they actually lie probably in one of our eye enhancers for PAX6. So it's probably likely that this genome-wide association study is pointing to variants in enhancers that control PAX6 expression. So we're now checking out these different SNPs, these different alleles at this locus in Shipra's zebrafish assay. Okay, so now I want to turn on to the next question about enhancers is what do they look like? What makes them special? Apart from the fact they bind transcription factors, is there anything else unusual about them? So first of all, we're going to look at something about the chromatin structure of enhancers themselves. And the work, this is the work of Pradeep Madhapura, who was a postdoc in my lab several years ago. He's now a new lecturer at the Blizzard Institute in London. So many groups around the world try and identify enhancers based on epigenetic marks that these enhancers carry. And most typically, people look at this modification here, H3K27, which is on the tail of the histone H3. This modification works by providing a binding site for effector proteins that bind that modification and influence gene regulation. So that's very well understood. And many groups have shown by chip sequencing that the H3K27 acetylation very nicely marks enhancers. So here's an example here. Here's the nanog locus. So this is data from mouse embryonic stem cells. The nanog locus, maybe it's better if I do this. The nanog locus here, here's one of its well-known enhancers here, and you see K27 acetylation there. So that's all good. So is that all there is to know about the chromatin structure of enhancers? And the answer is no. It's because almost the entire epigenetic field has been entirely focused on the tails of the histones that stick out from the nucleosome and bind these effector proteins here. But in fact, there are other places within histones that can get post-translationally modified and that lie right in the middle, the core of the nucleosome particle. So these modifications don't act in the same way as the tail modifications because proteins can't get in there and read those modifications. They act by actually physically disrupting the structure of the nucleosome itself, making it looser and probably contributing to gene activation by destabilizing nucleosome structure. So we decided to ask whether any of those modifications might be associated with enhancers. And working with Robert Schneider's lab in Munich, Pradeep concentrated on modifications, for example, of lysine 112, in histone H3, which sits really close to the DNA itself as it wraps around the outside of the nucleosome particle and is known to destabilize the nucleosome. So Pradeep did chip sequencing for H3K122 acetylation and also acetylation of H3K64, which lies close to the entry and exit point of DNA from the nucleus. And as you can see here, he found data that look very, very like H3K27. So enhancer nucleosomes not only have histone tail modifications, they've got modifications to the core of their structure. And moreover, Pradeep found that we could even find places that didn't have K27 acetylation, but did have modification, for example, of K122 acetylation and K64. And we published this several years ago. More recently, so again, 2 Indian postdocs in my lab, Dipta Semgupta and Yatendra Kumar, have been investigating if this is even more pervasive. And Dipta and Yatendra very recently discovered another modification, H3K115 acetylation. Again, modification very close to 122, almost in direct contact with the DNA that overlies the nucleosome. And this has a really striking profile. So here's the chip sequencing for 115. It's not present at these more distal enhancers here. It's limited to just this one enhancer of nanog. So there seems to be a really complex epigenetic code going on at nucleosomes that we're only just beginning to understand, and that now I think we have the tools, knowing about all these structures, to begin to get more mechanistic information about how they contribute to gene activation. And finally, to the last question, and I think probably the most interesting question in this field is, how on earth do these genetic elements work from such ridiculous genomic distances away from their target genes? How can a bit of non-coding DNA, million base pairs away from the gene it's supposed to control, stuck in the intron of a housekeeping gene, completely irrelevant to the developmental situation where that enhancer's got to work? How does it work? And so as a field, people have come to the conclusion that it probably has to involve some modification of chromatin structure at scale. And people have thought of various possible models. Early models suggested that enhancers might bind components of the transcriptional machinery, transcription factors, coactivators, even polymerase itself. And then perhaps they might move down the intervening chromatin fiber, kind of tracking until they ran into the promoter of the gene that they control. I think that's an easily understandable model, and I think there probably are examples of enhancers that work that way. But I would say most recently in the literature and in the field, in meetings, people have been discussing instead whether many enhancers, particularly those that are located at large distances from the genes that they control, that these enhancers might actually physically loop over to the gene that they're going to control and sit next to the target gene in space inside the cell nucleus, so that it wouldn't matter that there's a billion base pairs of DNA in between because the enhancer and the promoter are close together. Again, a really appealing model, and I'm going to present some data that suggests that maybe that's not the answer for all enhancers, or even many of them. So turning back to our model systems, I'm now going to really focus on the second locus here, the bottom one here, the sonic hedgehog gene. So sonic hedgehog encodes a very powerful embryonic morphogen that's going to be used at many times and places during mammalian development, embryonic development, including in the neural tube, in the brain, in the epithelia of the lungs and the gut, and most famously in the limb as well. And we know so far of about 15 enhancers that drive sonic hedgehog expression during development. In fact, I think a new one was just discovered the other week, so we're up to 16. I bet there's lots more. I bet there's at least 30, just like PAC6. So I suspect this large intergenic space, this 800 kb of non-coding DNA upstream of sonic hedgehog is probably packed full of sonic hedgehog enhancers yet to be discovered. Okay, so how does this regulatory landscape actually work? And sonic hedgehog's considered to be a real exemplar for this mode of long-range gene regulation. Because even though its enhancers are spread over such a large genomic distance, they are all contained within this red triangle here, this structure that's known as a topologically associated domain. So what's a topologically associated domain? So these data, these matrices here, are produced by methods such as chromosome conformation capture and high-C methods, molecular biology tools that allow you to query by DNA sequencing what DNA sequences might be physically close to other sequences in the genome. So what this triangle means is that all the DNA sequences in this bit of the genome, and this happens to be from mouse embryonic stem cells, prefer to interact with each other than they do interacting with the sequences in the adjacent triangle. Okay? So that triangle's a TAD, I've drawn it here, and so we imagine in our minds that maybe what it looks like is this is kind of a globular chromatin bundle here, that's a TAD, and then you move into the next TAD and that's a separate kind of bundle. How are these TADs formed? They're formed by a process called loop extrusion, so this is the work of many groups around the world, not our own, beautiful experimental biology. So loop extrusion is caused by the cohesin complex, a molecular motor, that's loaded onto the genome, traps DNA, and then moves along the genome until it hits a brick wall. And the brick wall is motifs for the zinc finger protein CTCF when it's in a particular orientation. So there, cohesin stalls and stops, so all the extruded DNA makes this topologically associated domain. Okay? And sonic hedgehog fits this very well, because the TAD boundaries there and there are marked by correctly orientated CTCF sites. So it all fits very nicely into the emerging model of this higher order, three-dimensional genome organization. So it's very suspicious and persuasive that all the enhancers for sonic hedgehog are contained within that TAD. Doesn't that suggest that maybe this structure is important for how these enhancers work at such large genomic distances? So let's explore that a little bit and address whether this structure is really important for the enhancers of sonic hedgehog being able to perhaps loop over and contact sonic hedgehog in space and time in development. So, and I'm going to focus first of all on the famous limb enhancer for sonic hedgehog, which was discovered by my colleague in Edinburgh, Bob Hill. It's called the ZRS. There it is there. Million base pairs away from the genic controls, 2 genes away from the genic controls. It's stuck in the intron of this gene called LMBR1, which is nothing to do with the limb. And the job of this enhancer is to activate the expression of sonic hedgehog for 2 days of mouse limb bud development in a very small patch of limb bud mesenchymal cells at the distal margin of the limb. So not in the proximal part of the limb, but the distal, and just in this tiny quadrant, a patch of cells on the posterior margin of the limb, which is called the zone of polarizing activity by embryologists. So somehow this element over here is integrating really complicated signaling environment going on during limb development, and is only being activated just in this patch of cells. And yet it's performing that really complicated operation and activating its target genes from 1 million base pairs away. Completely amazing. And this system is really easily perturbed because point mutations in this conserved limb enhancer for sonic hedgehog in humans, mice, 6-toed cats, and chickens as well. I think that picture of the chicken's gone wrong here, never mind. All produce the same phenotype of preaxial polydactyly, so that's too many digits and incorrect anterior posterior specification. So your thumb is your anterior digit. And each of these mutations, they're all in different places, but each of them creates an extra binding site for an ETS family transcription factor. And we know that ETS family transcription factors are important for limb development. So these are all gain-of-function mutations. It's making the sonic hedgehog limb enhancer too good. And here in this transgenic that was done by Laura Lettis in Bob's lab, you can see for one of these point mutations modeled in the mouse, that what it does is it expands the zone of sonic hedgehog expression on the posterior margin of the limb. So that's going to give you too many digits. And you start to fire off patches of expression on the anterior margin of the limb, which is going to mess up anterior-posterior polarity. Okay. So how is this executed? So we decided to ask whether we could find evidence of this chromosome looping mechanism, that this limb enhancer coming and being placed spatially next to the gene it's going to control during mouse limb bud development, and perhaps only in the patch of cells where the enhancer is going to be activated. Is this looping part of the mechanism? And the way we decided to do this was just go look. So a great thing about a large complex genome like the mammalian genome is it's really amenable to light microscopy, particularly now that we have super resolution optical microscopy. So here we're using structured illumination microscopy. So we have a resolution limit of just under 100 nanometers here. So this was an experiment done by my postdoc Ian Williamson together with Bob Hill's lab. So what we did is we used fluorescence in situ hybridization on mouse limb bud tissue sections at day, I think we did this at day 11 and a half. And Ian made a probe here in red for the sonic hedgehog gene itself, and then he made a second probe over here in green for the sonic hedgehog limb enhancer. And then the experiment's really simple. You go through the mouse limb bud and you ask how close are the red and green spots to each other in the nuclei. And so over here, on the top here, this is looking on the wrong side of the limb. So this is the anterior margin of the limb, where in the wild-type situation, the limb enhancer won't be active in driving sonic hedgehog expression. And what we saw there was, for both alleles here, the red and the green were close together. You know, they're in the same topologically associated domain, after all, but they were separable. And on average, they were about 300 nanometers apart from each other. Whereas in the right part of the limb, in the zone of polarizing activity where we know the limb enhancer is active and driving sonic hedgehog expression, we could find both alleles very typically right on top of each other within the resolution limit of our imaging, such that, for example, here, 35% of alleles, the enhancer and the promoter, are within 100 nanometers of each other, and here, just over about 45% of alleles within 200 nanometers. So we took this there's clearly a very different three-dimensional genome organization on these two halves of the mouse developing limb bud, and we took this as reasonable evidence to support the popular model of chromosome looping. We thought this was good visual evidence that the sonic hedgehog limb enhancer loops around, sits next to the promoter of the sonic hedgehog gene, and that's part of the mechanism that drives gene expression. But this is only a correlation. It's supportive evidence, but it's not good enough. We need to prove that actually this chromosome looping is important for the limb enhancer to drive sonic hedgehog expression. So how do we do that? We decided we would try and break the topologically associated domain and see what happened. So we decided to do this by mutating the CTCF sites that create the boundary of this topologically associated domain. So this was done in a collaboration again with Bob Hill's lab. So I'm going to show you data just from one of these. So we've done each of the CTCF sites in this region. I'm just going to show you the data from site 1. Site 1 makes the left-hand boundary of this topologically associated domain, and it keeps sonic hedgehog, the target gene, in the same TAD as all its enhancers. So what we did, we used CRISPR-Cas9 to take out the CTCF site homozygously in mouse ES cells, and we also generated animals from those cells. So first of all, do we disrupt the structure as would be suggested by the loop extrusion model of TAD formation? And the answer is yes. So here's some triangle diagrams again from Hi-C. Here's the wild type diagram, and I've put this green dotted line to show you the TAD for sonic hedgehog. And here's the same data from homozygous CTCF1 deleted cells and animals. And here's the green line again, and I hope you can see there's an absence of red signals here. And if you can't see that here's a difference plot of the mutant against the wild type. So where you see blue, that's a loss of interactions. Where you see red, that's a gain. And you can see this great big blue stripe up the left-hand side here, which is the boundary, the left-hand boundary of this topologically associated domain being lost, and actually the new boundary being formed here at site 2. And the important thing here is site 2 is the wrong side of sonic hedgehog. it now takes sonic hedgehog out of its own topologically associated domain and puts it instead in the topologically associated domain associated with these genes over here, which are not expressed in the limb. So this work was the work of Lauren Kane, who's a PhD student in my lab, and this work's on the bioarchive if you want to see it in more detail. The chromosome conformation capture suggests we've disrupted structure. Have we disrupted that enhancer promoter loop in the limb by doing this? And the answer is yes. So here's some more imaging. Again, this is the posterior limb bud again. I showed you the close spatial proximity of sonic hedgehog and its enhancers in there. And here in limb bud cells from homozygously CTCF1 deleted mice, we can see increased separation of the probes. So that high frequency of alleles where we see the enhancer, the promoter right on top of each other in the wild type limb bud, specific to the posterior and not the anterior, is greatly reduced in the CTCF1 deleted mice. So the frequency of co-localization is now down to that you'd see on the anterior margin of a wild-type limb. So that's all good. It looks like we have perturbed, indeed, this structure that we thought might be important for sonic hedgehog to be activated by the limb enhancer ZRS. So what have we done to sonic hedgehog expression in the limb? Any guesses? The answer is absolutely nothing. So there's a lesson for you. You can convince yourself by making correlations between structure and function that everything fits everyone's favorite model, but until you go and do the experiment and try and break it, you can't make a conclusion. So here we've clearly disrupted this structure, and we've had no impact whatsoever on the ability of the sonic hedgehog limb enhancer, 1 million base pairs away from the sonic hedgehog gene, to activate sonic hedgehog expression in the right place of the developing mouse limb bud. Moreover, I showed you all those other neural enhancers for sonic hedgehog. This is the expression of sonic hedgehog in the brain of wild type and CTCF1 deleted mice. So again, sonic hedgehog's in the wrong TAD. It's not in the same TAD as all its neural enhancers, and yet brain patterning of sonic hedgehog expression is absolutely fine. Moreover, these mice are fertile, they're born at Mendelian ratios, they're running around with very nice limbs with the right number of digits on them, so we cannot detect a developmental phenotype. So we might be able to see a loop, but actually our evidence suggests it's not important for function. Moreover, as we started to explore other sonic hedgehog enhancers, I think there's probably too much data on here for you to see, but this is looking in the neural tube, in neural tube development where sonic hedgehog expression is driven down in this ventral portion here. We actually found that in the expressing cells, the enhancers were further away from sonic hedgehog than they were in non-expressing cells, completely opposite of what you expect from a looping model. So what on earth is going on? And the answer is nobody knows, actually. So we've still got these existing models of how enhancers might work. Yes, there might be some enhancers that work by linking. Yes, there probably are enhancers that work by three-dimensional chromosome looping. But in our assays, it looks like that many enhancers don't fit any of these models. And in fact, the field has undergone a really rapid transformation in the last year, and the whole transcriptional field is now really rethinking gene regulation for a much more biophysical model rather than these kind of stereotypical chromosome architectures, which has arisen from the observation that many components of the transcriptional machinery undergo this biophysical transition in nuclei, which is called liquid-liquid phase separation. And they make these condensates with very high levels of factors trapped within them. And so the emerging model for enhancer function actually is there's no need for an enhancer to be physically juxtaposed next to the gene it's going to control. It just has to get within they both have to get within proximity of the same liquid phase separated condensate in order for mass action and just old-fashioned biochemistry to actually activate gene expression. So it's a really interesting model. It's very dynamic, regulatable, much more plastic, I think. I think it's really exciting. It's really fiendishly difficult to interrogate these kind of biophysical states in vivo in development. But that's the direction we're going in. So I just want to end there by thanking the people in the lab who've done the work I've talked to you about. So as I said, the first work that I told you about, understanding the pathogenicity of non-coding mutations is a work led by Shippa Bhatia, who's a fellow funded by the charity New Life. The work on histone modifications at nucleosomes associated with enhancers was initiated by Pradeep, who's now in London, and is being carried on by Yatendra and Dipta. And then the work on the sonic hedgehog limb enhancer that I told you about has been a collaboration with Bob Hill's lab, and it's been done by a shared PhD student, Lauren Kane, and a postdoc in my lab, Ian Williamson. And Sheila has been my research technician for a quarter of a century, can you believe it? And she's the most wonderful imager. So thank you very much for listening, and I hope I've got time for a few questions.
Thank you, Wendy. We probably have time for a couple of questions.
So, great talk, Wendy.
Thank you.
I was just wondering about the last part, talking about the proximity of gene loci. Even the physical model would predict that the loci would come together.
Well, close.
I mean, the distances you talk about are optical,
Yeah.
resolvable. And that would, I mean, the phase-separated regions would be much closer.
Well, so the condensates are not our work. They come from Tijan's lab, Xavier Darsac and Rick Young. And actually, they've shown that the condensates are up to about 300 nanometers in diameter. So that's about the separations we're seeing. So actually, I think the distances do work.
But still, how would you get connectivity? Because the condensates are fairly, I mean, I'm not saying that the condensate model is wrong, but I'm just saying your data doesn't support the condensate model.
Well, I think it's one thing that's missing in the condensate model is no one knows what the chromatin is doing. So people have only looked at the protein. So I think that's what's missing. And actually, I was talking to Ibrahim Cissé the other day, and he was saying, actually, it might not be the condensates per se that's important, but of course a condensate will have around it a zone of very high concentration as well, of the molecules diffusing in and out of the condensate. So actually, maybe the enhancer, the promoters, only need to get into a zone around the condensate. So I think the kind of zone of activity might be relatively large. So I don't think our data necessarily disagree with the condensate model at all.
Hi, Wendy. A very nice talk and very easy to follow. I have a question in relation to the last part as well. So as you know, cells are mechanosensitive entities, and the application of forces on the nucleus changes gene expression in physiology and pathology. I was wondering if the formation of these topologically associated domains might be influenced by the application of forces on the nucleus.
That's a really interesting question. I don't know if anybody's done an experiment. It would be really interesting. Look, my favorite model to study that is the macrophage going through the endothelium, where the nucleus goes from being round to being a rod. So you'd imagine there must be massive changes in 3D structure. But as far as I know, no one's ever addressed that. Really interesting question.
We'll take one last question,
Okay.
Nice talk,
and
Wendy.
then we'll go to the next
Yeah,
session.
Dimple's got one.
Very nice talk. I was wondering, since now, once you deleted the CDCO boundary,
Yeah,
and you see the TAD is shifted towards right,
yeah.
maybe now SHH is in control of another enhancer in the neighboring TAD, and so you don't see gene expression
No,
changes.
we know by genetically that the only enhancer capable of driving sonic hedgehog expression is the limb enhancer. There's no other enhancer in the genome that can activate sonic hedgehog in the limb. That's
Even
quite
if the
clear.
TAD is now SHH in a different TAD, still you think that another enhancer would not control
No.
the gene?
And now sonic hedgehog's in the adjacent, it's in the left-hand TAD now, where all the enhancers for Engrailed and Canopy 1 are, and we see no activation of sonic hedgehog by their enhancers when it moves into their TAD. So we can't see any change in spatial patterning of gene expression. We don't know whether there's a quantitative difference in the levels of expression, so we're exploring that at the moment.
Thank you. Thank you, Wendy, for the talk. If there are any more questions, we can have them later over tea.